Swayam Prabha. Digital India. Educated India.
Welcome to this 14th lecture in this course on eukaryotic gene expression. Today we are going to discuss about the regulation of biosynthesis of tRNA and fibers ribosomal RNA by RNA polymerase III. Now, the last 12 lectures or last 13 lectures, our focus has been primarily on the regulation of gene expression by RNA polymerase II, which we took about 12 lectures where I described clearly how RNA polymerase II along with general transcription factors and a variety of transcription factors which bind to upstream promoter elements, enhancer sequences, etc., go and bind to their corresponding response elements in the promoters and activate transcription, how differential gene expression takes place with the combination of core promoter elements, combination of variety of general transcription factors as well as sequence specific DNA binding proteins binding to upstream enhancer sequences and silencers and so on and so forth. We also discussed extensively about the role of chromatin in the regulation of gene expression, how histone modification, DNA methylation, etc. affect gene regulation, and then we discussed considerable amount of time discussing after transcription initiation how RNA processing events take place such as mRNA capping, mRNA splicing, polymerization and so on and so forth, and very importantly we discussed about the role of RNA polymerase C terminal domain in all these RNA processing events. And after discussing all these processes with relation to RNA polymerase II transcription, we spent the last lecture we discussed primarily about the regulation of ribosomal RNA synthesis by RNA polymerase I. Today we are going to discuss about the regulation of RNA polymerase III transcription, especially the regulation of tRNA and fibers ribosomal RNA synthesis by RNA polymerase III. So this is going to be the focus of today's lecture. RNA polymerase III, as we have seen in the case of Pol I or Pol II, is also a multi-subunit complex, and in fact there are about 17 genes which code the various subunits of RNA polymerase III. So it is a multi-subunit complex, it is a huge protein complex of at least 17 different subunits. Of these 17 subunits, 5 of the subunits constitutes the core subunits of RNA polymerase III. And these core subunits, most of these core subunits have homologs in both Pol I and Pol II. That means these subunits are kind of shared by Pol I and Pol II, but they have homologs. For example, the C160 subunit of RNA polymerase III is a homolog of Pol I 190 subunit as well as Pol II 220, which is the largest subunit of RNA polymerase II, and is kind of homolog of bacterial beta prime subunit of E. coli RNA polymerase I or RNA polymerase. Similarly, the C128 subunit of RNA polymerase III is a homolog of Pol I 110, Pol II 150 and the bacterial beta subunit of E. coli RNA polymerase. The C40 subunit of Pol III is also referred to as AC40 because the same subunit is shared by both polymerase I, which is referred to as Pol A, and polymerase III, which is often referred to as polymerase III. So it is called AC40 because this 40 kDa subunit is shared by both polymerase I and polymerase III and is common to both Pol I and Pol III. In the case of Pol II, its homolog is B45, Pol II B45 also known as RPB3, and it is also the bacterial E. coli or E. coli RNA polymerase alpha is the homolog in the case of prokaryotes. Similarly, the AC19, so called because this subunit is shared by both Pol I and Pol III, and the homolog in the case of Pol II is the Pol II B12.5 or also known as RPB11 subunit in the case of Pol II. Similarly, the ABC23 is shared by all the 3 RNA polymerases, namely Pol I, Pol II and Pol III, and it is known as RPB6 in the case of RNA polymerase II and is the equivalent of bacterial omega subunit. So this has been very brief the subunit structure of RNA polymerase. We will not go into extensive details of how RNA polymerase II subunit is organized, the structure and so on and so forth, but suffice to know that these 5 subunits actually constitute the core RNA polymerase III and they have homologs in case of Pol I and Pol III. Now, RNA polymerase III transcribes a set of genes whose main common features are that they encode structural or catalytic RNAs and in general they are shorter than 400 base pairs. So RNA polymerase III gives rise to RNA molecules which are approximately 400 base pairs or shorter than that. Okay. And what are these kinds of RNA molecules which are made by RNA polymerase III? These include transfer RNAs, 5S ribosomal RNA, U6 spliceosomal RNA, RNase-P and RNase MRP RNA. We will discuss each one of them in detail as we go along in this lecture series. So, I am just making a list here. We will discuss each one of them, but I will describe each one of them at the end of the lecture. But in today's topic, primarily we are going to discuss about the synthesis of 3 RNA species by RNA polymerase III, namely regulation of transfer RNA synthesis, mechanism and synthesis of 5S ribosomal RNA and U6 spliceosomal RNA. How are these 3 major RNA species are actually synthesized by RNA polymerase III? What is their promoter organization? What kind of transacting factors are involved, or which coordinate the RNA polymerase III leading to synthesis of these 3 major types? The others I will just only make a passing note on it, because it is not possible to cover everything. So, let us now try to understand how RNA polymerase III synthesizes transfer RNA, 5S ribosomal RNA and the U6 spliceosomal RNA. One of the very unique features about RNA polymerase III, which we have not found in case of polymerase II or polymerase I promoters, is that the RNA polymerase III promoters can be either extragenic or intragenic. Whereas so far all our discussion in the last 13 lectures, which centered around RNA Pol II transcription, Pol I transcription, the promoter always lies towards the 5' end from the transcription start site. So, they are almost referred to as the extragenic promoters. They do not, the promoter lies 5' to the transcription start site and not within the coding region. It is only in the case of Pol III transcription, some of these Pol III transcription promoters or Pol III transcript promoters, the promoter can be intragenic, that means within the transcribed region. It can also be extragenic. So, this is one of the fundamental differences between Pol III transcription or promoters transcribed by Pol III compared to Pol I and Pol II promoters. The intragenic promoters are generally Tata-less, that means they do not contain a Tata box, while the extragenic promoters generally contain a Tata box. So, this is another important difference between the extragenic and intragenic promoters. So, let us just take a bird's eye view of what are these 3 different Pol III promoters, what are the major elements, and then we will take each one of them and discuss in greater detail. So, basically depending upon whether the promoter are intragenic or extragenic, the Pol III promoters are of 3 major types. They are called type 1, type 2 and type 3. The best example for the type 1 promoters are the promoters of genes encoding 5S ribosomal RNA. Remember, when we discussed about Pol I transcription and regulation in the last class, we had clearly discussed the RNA component of the ribosomes consists of 28S, 18S, 5.8S and 5S, and of which the 5S rRNA is actually synthesized by Pol III. So, that is what we are now going to discuss in detail now. The other 3 are synthesized by RNA polymerase I, and we have discussed very clearly, to get a functional ribosome you require the coordinate function of all the 3 RNA polymerases, namely Pol I, which makes 28S, 18S and 5.8S rRNA, and 5S rRNA, which is made by Pol III, and all the ribosomal proteins are made by messenger RNAs, which are transcribed with the RNA polymerase II. So, to get a functional ribosome, all 3 RNA polymerases have to work together. Today, let us now try to understand how the 5S rRNA gene is transcribed by RNA polymerase III, how is this promoter organized. So, it turns out this 5S ribosomal RNA promoter consists of, it is an internal promoter, like just previous slide we had discussed. The RNA Pol III promoters can be either extragenic or intragenic, or either can be external or internal with reference to the transcription start site. The type 1 promoters, which is an example we are going to discuss, the 5S ribosomal RNA promoter is actually an intragenic promoter, where the promoter lies after the transcription start site, and there are 2 key promoter elements which are very important for this transcription of 5S rRNA gene. They are called as box A and box C. In the same way, when you come to type 2, which all majority of the transfer RNA genes, majority of the genes encoding various types of transfer RNA fall under the category of again type 2 promoters. Again, this is an intragenic promoter because the 2 major promoter elements, namely the box A and box B, is within the transcribed region, lies 3' of the transcription start site. The type 3 we are going to discuss as an example the human U6 snRNA. Remember, the U6 snRNAs are components of the splicing machinery. The splicing machinery consists of a number of ribonucleoprotein particles, which are RNA plus protein components, and many of these RNA components are encoded by the Pol III RNA polymerase, and the U6 RNA is one such promoter we are going to discuss here, and this comes under type 3 because here the promoter is extragenic, because it is not within the transcribed region compared to other two. It lies in the untranscribed region over the 5' end of the 5' to the transcription start site, and there are three major elements, promoter elements in the case of this U6 RNA are the type 3 promoters, which consists of Tata box, which consists of another, and then there is a near upstream element called proximal sequence element or PSE, and then further away you have what is called as a DSE or a distal sequence element. So remember the Pol III transcription, the Pol III promoters are of three distinct types, type 1, type 2 and type 3. Type 1 and type 2, the examples are 5s rRNA and transfer RNA promoters, they are intragenic, whereas the type 3 promoters as exemplified by the human U6 snRNA is extragenic, since the promoter lies outside the transcribed region. Let us now discuss little bit in greater detail how is the Pol III promoter of 5s rRNA gene is organized, what are the key promoter elements. This belongs to type 1 and it is an intragenic promoter. It turns out the intragenic promoter in the case of the 5s rRNA gene consists of three major elements, which together consist of what is called as a internal control region, often referred to as ICR. Now the internal control region comprises of three distinct elements. It consists of what is called an A box, which I have shown in red color here, which spans from plus 50 to plus 64. So the transcription start site is plus 1. So the promoter lies after the transcription start site, that is why we call it as intragenic. So the A box spans from plus 50 to plus 64. There is immediately after that you have what is called say intermediate element, which spans from plus 67 to plus 72 with respect to transcription start site, and then there is what is known as a C box, which spans from plus 80 to plus 90. And it also turns out in the case of all the three RNA Pol promoters, namely the type 1, type 2 or type 3, whether it is extragenic or intragenic, the termination mechanism is more or less similar and is actually dictated by a stretch of trees. We will discuss in more detail about the transcription termination by RNA polymerase through later in this talk, but remember a simple stretch of T's, even 10 to 20 residues of T is good enough to terminate RNA Pol III transcription. And this transcription termination signal is common whether it is type 1 or type 2 or type 3 promoters. So transcription termination signal in the case of Pol III promoters primarily consists of a stretch of T residues ranging from 10 to 20 residues. So this is basically how a Pol III promoter of Xenopus laevis 5s rRNA gene is organized. Remember there are three major elements, a box A, box C, and in between you have what is called as an internal control, intermediate element. Together they constitute what is called as a internal control region. So these are the major cis-acting elements of a 5s rRNA promoter transcribed by RNA polymerase 3. Now let us look at the tRNA promoters. Here they are taken example of Xenopus laevis tRNA coding for leucine, the tRNA leucine gene, which belongs to the type 2 category. Unlike the 5s rRNA promoters where we have discussed the major promoter elements are box A and box C, here the key promoter elements are known as box A and box B. Again they are internal because the box A spans from plus 8 to plus 19, whereas box B spans from plus 52 to plus 62. So they come after the transcription start site, therefore they are known as intrinsic. And as observed in the case of the type 1 promoter or the 5s rRNA promoter, even here the transcription termination is dictated by a series of T residues which act as a transcription termination signal even in the case of the type 2 promoters. When you come to the Pol III promoter of human U6 srRNA gene, remember it is a type 3 promoter and it is an extragenic promoter where all the cis-acting elements are present towards the 5' end before the transcription start site, whereas in the other two promoters which we discussed so far, the promoters are internal. So the major cis-acting elements are what is called as a distal sequence elements, which usually lies about 200 base pairs away from the transcription start site and is often known as the DSE, and it also consists of a core promoter which there are two major promoter elements in the core promoter element, namely the proximal sequence element or PSE, which spans between minus 65 to minus 45 as shown in blue color here, and a Tata box which is minus 32 to minus 25. So the two intragenic promoters, 5s rRNA gene promoter and the tRNA gene promoter, do not contain any Tata element and the promoters are internal, whereas in the case of the human U6 snRNA gene, the promoter is extragenic and it contains a Tata element and two other promoter elements referred to as PSC and DSC. And as seen in the case of the introgenic promoters, even here a simple stretch of T residues towards the 3' end of the gene act as a transcription termination signal. So, irrespective of whether the promoters are introgenic or extrogenic, the transcription termination mechanism is more or less same. So, although I have described these 3 basic types of Pol III promoters, there are always variations, there are always exceptions to this rule. For example, in the case of Saccharomyces cerevisiae, the U6SNRNA gene promoter consists of cis-acting elements which are both extragenic as well as intragenic. So, although basically there are 3 types of Pol III promoters, there are variations, there are exceptions to this rule, and one such exception is what I have shown here, where the Pol III promoter of Saccharomyces cerevisiae U6SNRNA gene is a kind of a hybrid promoter because it contains a Tata box, so which is extragenic 5' to transcription sites, and it also contains a box A within the transcribed region, and it contains a box B at the 3' flanking region of the U6SNRNA gene. So, the Tata box is located from -30 to -23, whereas the A box is located from +21 to +31 within the coding region, whereas the B box is located far downstream of the U6 coding region from +234 to +244. So, there are variations from the 3 basic types, such as the Saccharomyces cerevisiae U6 gene, where it can be both extragenic as well as intragenic. Again, the transcription termination is brought about by a stretch of T residues as seen in the case of the other Pol III promoters. Now, how did people find out that the promoter in the case of the Pol III transcription unit is internal? As we have studied in the case of the Pol II promoters, primarily studies involving mutations, site-directed mutagenesis and deletions have helped people to demark what are the major cis-acting elements involved in the Pol III promoter. For example, I have just shown a cartoon here to illustrate one experiment which actually demonstrated that the promoter in the case of the 5S rRNA gene is actually internal. So, basically the strategy is similar to what we have studied in the case of the Pol III transcriptions, where when people started making deletions towards the 5' end of the transcription start site, people found out when you delete the 5' regions before the transcription start site, it had no effect on the promoter activity. The promoter activity was not affected when you make deletions 5' to the transcription start site, clearly indicating that there is no cis-acting elements or there is no major promoter elements 5' to the transcription start site. But the moment they made a deletion which spans after the transcription start site, there was a dramatic decrease in the promoter activity, clearly indicating that the promoter region actually is after the transcription start site, and these kinds of study gave a hint that yes, in the case of tRNA gene promoters, unlike what people have seen in the case of Pol II and Pol I, here the promoter may be actually internal. Similarly, people have started making deletions from the 3' side, and again there was no effect as long as you delete different regions of the 3' region, but the moment you started making deletions spawning the internal control region, there was a dramatic decrease in the promoter activity, clearly indicating that these kinds of studies, when by doing deletion studies from both sides, people have been able to identify an internal control region which spans from which spans after the transcription start site. This is how the internal control regions or the internal promoters of Pol II transcription genes were identified. Now, so far we have discussed about the major cis-acting elements which control RNA polymerase III transcription. As we discussed, you have type 1 promoter, you have type 2 and type 3 promoters, and a typical example for type 1 promoter is the 5S ribosomal RNA gene, which contains box A and box B, and you have the type 2 promoters. Most of the tRNA genes fall under this category. They have the box A and box C, and then we discuss about extragenic promoters in the case of the human U6SNRNA, where the promoter is external or extragenic, which contains of Tata box, a proximal sequence element and a distal sequence element. And there are variations as seen in the case of the Saccharomyces U6SNRNA gene. Now, let us spend some time to understand what kind of transacting factors go and interact with the cis-acting elements. It turns out there are 3 major transacting factors which govern the regulation of transcription by RNA polymerase III. These are known as TF3A, TF3B and TF3C. Remember, whenever we talked about Pol II transcription, the designation has been TF2. All the factors were designated as TF2A, TF2B, TF2C and so on and so forth. Whereas when it came to Pol I, they are designated as TF1. Whereas when it comes to Pol III, you use the terminology TF3. So, TF3 means the factor is associated with Pol III transcription, TF2 means the factor is associated with Pol II transcription, TF1 means this transcription factor is associated with Pol I transcription. It turns out the type 1 promoters, as discussed in the case of the 5' ribosomal RNA, requires all these 3 transcription factors, namely TF3A, TF3B and TF3 in addition to RNA polymerase III. Whereas the type 2 promoters, which primarily consists of a majority of tRNA gene promoters, they require only TF3C and TF3B, they do not require TF3A. So, the TF3A is very specific for the transcription of 5' ribosomal RNA gene and is not required for the transcription of the transfer RNA genes. Now, what kind of experiments people have done to actually demonstrate that TF3A is required only for the 5' ribosomal RNA promoter, but not for the transcription of tRNA genes. As we have seen in the case of the Pol II transcription, people have again made use of cell-free transcription studies. In fact, the Pol III transcription studies and Pol III cis-acting elements and transactive factors were identified much before the Pol II transcription studies were initiated. In fact, the TF3A was the first transcription factor, eukaryotic transcription factor, to have been purified to homogeneity and studied very extensively. In fact, it is one of the first zinc finger proteins which were ever identified. So, let us say for example, I have shown you an experiment, a simple cell-free transcription studies where you put all the transcription factors, namely the RNA polymerase III nucleotides, and you put a 5' RNA promoter as well as a tRNA promoter linked to the downstream gene, and then see what kind of factors are required for the transcription. And it turns out when you do not have TF3A, how do you deplete the extracts of TF3A? You simply add antibodies raised against TF3A, and these antibodies raised against TF3A will prevent the TF3A from interacting with its cis-acting element and therefore makes unavailable for the cell-free transcription. So, when you make the TF3A in a cell-free extract non-functional by the addition of an antibody, you can see the 5' RNA gene transcription is totally gone. But in the extracts depleted of TF3A, tRNA transcription still proceeds. These two are controls where you have not had any antibody, you have added a non-specific antibody, but here you have added a specific antibody raised for the which are specific for TF3A. So, when TF3A function is inactivated, only 5' ribosomal gene transcription gets affected, but not transfer RNA, clearly indicating that the TF3A is a highly specific transcription factor required specifically for the transcription of 5' ribosomal RNA genes. So, these kinds of experiments have clearly demonstrated which of these 3 transcription factors are required for 5' RNA gene transcription as well as tRNA gene transcription. I have just described here a cartoon which actually depicts the structure of the TF3A zinc finger protein. The TF3A consists of 9 zinc fingers, and again I will not go in very detail about the zinc finger proteins because we have studied extensively in the RNA polymerase II lectures. In fact, if you want to know more about the zinc fingers, you must go to the lecture number 5, where we have discussed in detail about the C2H2 and kind of zinc fingers and other kind of zinc fingers in great detail and giving examples of Pol II transcription factors. So, the TF3A consists of 9 such zinc fingers, where a zinc atom is coordinated between 2 cysteines and 2 histidines, and it's a C2H2 kind of a zinc finger module, and it consists of 9 tandem sequences that are very closely spaced and together constitutes the 9 zinc finger motifs which go and interact with the 5' internal control region of the 5' RNA promoters, and the consensus amino acid sequence is shown here. And as I said, if you more discussion about zinc fingers, we need to go back to lecture 5 and then come back here and then try to understand how the zinc atom is coordinated and what are the major structures of the major features of the zinc finger proteins. As I mentioned, there is extensive literature on the TF3A. In fact, TF3A is one of the first eukaryotic transcription factors which are purified to homogeneity, and there are a number of factions. I listed here a number of review articles in the early and late 1990s which discuss about the discovery and various structural and functional aspects of TF3A. So, you have to refer to some of these articles if you want to learn more about TF3A, and it is a very interesting protein and how it binds to internal control region and allows RNA polymerase to go through. There are very interesting mechanistic studies which have been studied to understand how TF3A binds and how RNA polymerase III transcribes the 5' RNA genes. We will not go into the details now. So, transcription initiation of the Pol III promoter involves the assembly of TF3A and TF3E, TF3C, and the TF3B, as I said, TF3A is a zinc finger protein, and in addition to TF3A and TF3C, it requires the initiation factor TF3B. All these are multi subunit proteins. So, remember they are not simple proteins. For sake of simplicity, I have simply mentioned it as a single protein, but these are all multi subunit proteins. For example, the TF3B consists of Tata binding protein, BDP1 as well as BRF1. Again, we will not go into the details of subunit structure of all these transcription factors and their interactions and so on and so forth. Suffice to remember that 3 major transcription factors are required for the transcription of 5S ribosomal RNA gene, namely TF3A, TF3B and TF3C. But in the case of tRNA gene transcription, you require only 2 factors, namely TF3B and TF3C. Of course, RNA polymerase is required in addition to all these 3 transcription factors, and assembly of all these 3 factors along with RNA polymerase completes the pre-initiation complex formation in the case of Pol III transcription. So, again, as we seen in the case of the polymerase II transcription, where the TF2D and the TF2A come and occupy first, and then the RNA polymerase is brought in and other factors come and join and the pre-initiation complex is formed. The same way, ordered sequential binding of transcription factors to the 5S RNA gene results in the formation of a pre-initiation complex. In the case of Pol III promoters, for example, the internal control region of the 5S RNA gene is first bound by TF3A. So, TF3A is the first transcription factor to go and bind to the internal control region, but this results in the formation of very unstable internal control region TF3A complex. This DNA protein complex is stabilized by the joining of TF3C. So, you get a very stable DNA protein complex involving internal control region TF3A and TF3C, and once this stable complex is formed, then the TF3B is recruited and you get a very stable pre-initiation complex, and then the Pol III is recruited, and in presence of nucleotides it makes the 5S ribosomal RNA gene and RNA transcription termination takes place, and then the cycle continues. So, TF3A is recruited first, TF3C is recruited next, followed by TF3B and RNA polymerase, and all of them assemble together to synthesize the Pol III transcription unit. So far we discussed about the transcription factors binding to the internal control, introgenic promoters, namely the 5S rRNA and tRNA genes, where you have the TF3A in the case of the 5S ribosomal RNA gene, and you have, in addition to TF3A, also have TF3C and TF3B in the case of the tRNA genes. So, let us very briefly discuss about the various transacting factors which are involved in the regulation of in the extragenic promoter, namely the human U6 snRNA gene, where you do not have either box C or box B or box C, but you actually have extragenic promoters in the form of a Tata box, a proximal sequence element and a distal sequence element. So, what kind of protein factors go and bind to this? The transacting factor which bind to the U6 snRNA promoter in the case of humans, where the promoter is extragenic, are primarily an OCT1 and a staph, ZNF, ZNF or SBF protein. These are variously named. They bind to the distal sequence element, whereas a protein called SNAPC or the snRNA activated protein complex, also known as the PTF or the proximal sequence element associated transcription factor, binds to the proximal sequence element, and of course the Tata binding protein occupies the Tata box. So, these are the major transacting factors which interact with the cis-acting elements of the human U6 snRNA gene, where the promoter is extragenic, whereas in the case of the other two, namely the 5S rRNA gene, you have TF3A, TF3B and TF3C, as in the case of tRNA genes it is TF3B and TF3C. So, these are the cis-acting elements and transacting factors involved in the regulation of Pol III transcription. As I said, I have made a very simplistic view of Pol III transcription. It is much more complicated than what I have discussed just now. The idea is not to confuse the viewers and not to confuse the learners, but to give a very overall view of the mechanism by which the Pol III transcription takes place. So, I have given a very bird's eye view of the major cis-acting elements and transacting factors involved in the Pol III transcription. You want to study more details about the Pol III transcription, there is an excellent review in Genes and Development in 2002 by Schramm and Hernandez about the recruitment of RNA polymerase through target promoters, how all these transacting factors, TF3A, TF3B and TF3C interact with each other to recruit the RNA polymerase together, and then how RNA polymerase actually transcribes and how the transcription termination takes place and so forth. So, for more details you should read review articles such as this. There are a number of other review articles, but this is one of the very interesting and very elaborate review. Now, as I said, termination of Pol III transcription is very simple, and it is actually determined by a short stretch of U residues in the case of RNA, or T stretch, which is corresponding the T residues in the case of the genes. So, a very short stretch of U residues can lead to transcription termination in the case of the RNA polymerase III, and this is one of the reasons why polymerase III actually produces very short transcripts, less than 400 base pairs, like tRNA, 5S RNA, and so forth, so on, so forth. For example, in the case of the U4SN RNA, the and human Pol III, the U4 to U5, the just 4 residues of U is good enough to bring about transcription termination in the case of the human and Xenopus polymerase III, whereas in the case of Saccharomyces cerevisiae polymerase III, about 4 to 5 residues of T's are used good enough to bring about transcription termination, whereas in the case of Saccharomyces cerevisiae Pol III, about 5 to 6 residues of T in the gene or U residues in the RNA is good enough to bring about transcription termination. And it turns out, of the various subunits of RNA polymerase III which we discussed earlier, the C11 subunit of RNA polymerase III is actually essential for the termination, since if the C11 subunit is deleted or mutated, the termination efficiency decreases strongly. So, of the various RNA polymerase II subunits, I think about 15 or so, the C11 subunit is actually responsible for transcription termination. So, the C11 subunit of Pol III is actually as the transcription termination factor. So, remember, transcription termination in the case of Pol III is very simple. It simply consists of a stress of series of T residues which can range anywhere from 4 to 6 depending upon the species, whether it is Saccharomyces cerevisiae, human, or other species. It looks very extensive, but the purpose is not to scare you, but just to tell you, in addition to the 5S ribosomal RNA, tRNA, and the U6 RNA, the RNA polymerase III also transfers a number of other RNA. As a number of other molecules. Many a times you don't know. Whenever we ask you a question, usually the students say Pol III transcription primarily means transfer RNA genes, but it is not true. In addition to the transfer RNA genes, RNA polymerase III also transcribes 5S ribosomal RNA. Many of the RNAs involved in RNA splicing, such as U6 and so on and so forth. It also makes a number of RNAs, which I will. So, let us spend maybe a few minutes to understand what are these various RNA molecules, what are their biological functions, and why these RNAs are also being synthesized by RNA polymerase. For example, in addition to the tRNAs, in addition to the 5S ribosomal RNA and the U6SN RNA, it also makes all these RNAs. The RNase P RNA is actually, its RNase P is an endoribonuclease that processes the T prime, the 5' terminal of the P tRNAs. So, ribonucleases, which is a ribonucleoprotein, RNase P is a ribonucleoprotein, and RNA component of the ribonucleoprotein, which is actually required for the tRNA processing, is actually synthesized by tRNA, by RNA polymerase III. Similarly, there is an RNA called mRpRNA, which actually refers for RNAs involved in mitochondrial RNA processing, which is an RNA dependent endoribonuclease that processes RNA primers for DNA replication in mitochondria, and is also present in nucleolus where it requires in pre-RNA processing. So, this mRpRNA is also made by RNA polymerase III. And there is a very important RNA called 7SL RNA, which is actually is a part of the signal recognition particle, which is very important for the bringing the nascent polypeptide chains to the rough endoplasmic reticulum and channelizing proteins towards the secretory pathway. So, all the secretory proteins which contain a signal sequence, they are brought about during translation. They are translated on rough endoplasmic reticulums, and a key protein called a signal recognition particle plays a very crucial role in this process, and the signal recognition particle is a ribonucleic protein, and the RNA component of the signal recognition particle is actually made by RNA polymerase III. Similarly, you have 7SK RNA, which actually binds to the transcription elongation factor PTFB and is involved in repression of Pol II transcription, and this regulatory RNA is also made by RNA polymerase III. Similarly, there are what are called as Walt RNAs, which are large cytoplasmic ribonucleoproteins implicated in multi-drug resistance of human tumors and are thought to be involved in macromolecular assembly and transport. So, the RNA components of these ribonucleoproteins are also made by RNA polymerase III. And there are what are called as yRNAs, which are very small RNAs of the ribonucleoprotein RORNP, which is the target of autoimmune antibodies in patients with an autoimmune disorder called systemic lupus erythematosus. So, you can see Pol III is not just involved in transcription of tRNA and 5' RNA, it also seems to be involved in synthesis of a number of other important less known RNAs, but they are very important. Similarly, in the case of nervous system, you have what are called as BC1 and BC200 RNAs, which are RNAs very specifically expressed in neural cells, and they form what are very important components of cytoplasmic ribonucleoproteins, and they play a very important role in the translation of dendritic messenger RNAs in the case of nervous system. So, neural cells. Similarly, there are what are called as virus encoded RNAs VA1 and VA2, which act by inhibiting the RNA activated protein kinases in response to viral infection, thus allowing the translation of adenoviral mRNAs. And there are what are called as SINEs, short interspecific repeat DNA elements encoded RNAs. These are non-autonomous retroposons originating from Pol III transcribed genes. I have made this particular list just to tell you that tomorrow if somebody asks you what is the function of RNA polymerase III in eukaryotic cells, do not say that it simply makes transfer RNA, you have to say it makes transfer RNA, primarily transfer RNA, 5' ribosomal RNA and U6 RNAs involved in splicing, but it also makes a number of other less known RNAs, some of which I have listed here. So, Pol III has very important functions inside the cells. So, just to recapitulate your me, what we have discussed so far I have just summarized here. Synthesis of transfer RNA by RNA polymerase III machinery involves the use of promoter elements located within the transcribed region of tRNA genes. So, the tRNA gene promoters, the promoters are internal. In most tRNA genes transcribed by RNA polymerase III, these internal promoter elements are known as box A and box B. So, all tRNA gene promoters are internal, they contain two important elements called box A and box B. The transcription factor TF3B and TF3C are required for the initiation of transcription by Pol III promoters of tRNA genes. So, if somebody asks you describe a tRNA gene promoter and the transacting factors, it is very simple. The tRNA gene promoters are internal, they consist of two major cis-acting elements box A and box B, and two transcription factors called TF3B and TF3C together with RNA polymerase III are involved in transcription initiation in the case of the tRNA gene promoters. Whereas, if you come to 5' RNA, it is slightly more complicated than tRNA gene promoters, where the cis-acting elements instead of box A and B are designated as box B and box C, and in addition to TF3B and TF3C, they also require TF3A. So, three major transacting factors are required for transcription of 5' RNA gene promoters, whereas only two transacting factors are required for the transcription of tRNA gene promoters. This is the major distinction between the two. The TF3B basically recruits the RNA polymerase III and promotes transcription initiation as well as reinitiation. And one of the components of TF3B is the TATA binding protein, and also in the case of Drosophila melanoic acid, there is a protein called TBP related factor or TRF1, and these actually interact with the TATA like sequence, which is usually present around 30 region from the transcription start site in many of the Pol III promoters. So, when you have an extragenic promoters, you require a TATA binding protein which binds to a TATA sequence. So, in addition to the TATA box, that is in the case of extragenic promoters, in addition to TATA box, you require two major elements, namely the prox segment sequence element located about 20 base pairs upstream of TATA box, and a protein called SNAPC, also known as PBP or PTF, binds to this proximal sequence element. And another element is the DSC or the distal sequence element located about 20 base pairs upstream of the TATA box, and it binds to transcription factors such as STAF or OCT1 and further enhances the rate of polymerase transcription. Okay. So, I have type 1 promoters, the reference by tRNA, which requires box A and box B, and you require two transcription factors TF3B and TF3C. You have 5' RNA promoters, you have box A and box C, which are occupied by TF3B, TF3C, and also a unique transcription factor called TF3A, which is zinc finger protein. Whereas, when you come to extragenic promoters, there are three major cis-acting elements, the TATA box, proximal sequence element and distal sequence element, and these are occupied by distinct transcription factors. So, this is in a nutshell the organization of polymerase III promoters and how RNA polymerase III transcribes Pol III promoters, tRNA, 5' RNA and U6 RNA and various other promoters using specific transcription factors. So, the polymerase III is recruited by the promoter by TF3B, and the TF3C is composed of C subunits, and it recruits TF3B to the DNA sites upstream of the transcription start site. So, as I said, each one of them is a multi subunit complex. Do not think it is as simple as what I mentioned here, it is much more complicated. TF3B, TF3, each one of them is a multi subunit complex, and each subunit has a very important regulatory role, and the TF3A, which is the 5' rRNA gene specific DNA binding protein, it forms the platform for binding of TF3C to these genes. So, if somebody asks you what is the function of TF3A, you have to mention TF3A is a very important transcription factor required for the transcription of 5S ribosomal RNA genes. So, that is the summary of what we have discussed so far. Now, we will spend about maybe 10 minutes or so now to discuss what is the physiological significance of Pol III transcription. As I said, in general, people tend to concentrate primarily on RNA polymerase II transcription, how protein coding genes are transcribed, how messenger RNA is synthesized. Most of the time the focus is primarily on the TF2D, TF2B, RNA polymerase II, CTD, splicing, capping, polyrylation and so on and so forth. Very rarely people describe Pol I and Pol III transcription. So, in the last class when I discussed Pol I transcription, I mentioned very clearly why it is important to understand the rDNA transcription and how rDNA transcription plays a very important role in cell growth, and in many of the cancers the Pol III transcription is enhanced, and you do require both Pol I transcription, Pol III transcription, because if the cell has to grow and divide, proteins have to be made, and if proteins have to be made, you require ribosomes and you require transfer RNA, and therefore these two transcriptions are also very important from the point of cell physiology and function. It turns out, just as we have seen in the case of the RNA polymerase I transcription, where it plays a very important role, and in many cancers the Pol I transcription is either enhanced and many of the Pol I transcription factors are overexpressed. In the same way, the Pol III transcription also plays a very important role in cancer. Let us now try to understand how it is. We all know protein synthesis is a prerequisite for rapid cell growth. So, if cells have to grow and divide, proteins have to be constantly made. The growth rate is directly proportional to the rate of protein accumulation, and it turns out if the protein synthesis is reduced by about 50%, the cells withdraw from the cell cycle and go into a quiescent state. So, the cells have to continuously divide, the protein synthesis has to take place continuously, and if synthesis has to take place continuously, since the ribosomes have to be made, tRNAs have to be made, that means Pol I transcription and Pol III transcription have to be regulated continuously. And if protein synthesis falls 50% of the normal, the cells will exit the cell cycle and go into a quiescent state. This is the basic principle. Now, the availability of transfer RNA and ribosomal RNA therefore is a very important determinant for the rate of translation. So, if protein synthesis is very important for the cells to grow and divide, the protein synthesis rate is controlled by Pol I and Pol III transcription. High levels of Pol III transcription are therefore necessary to sustain rapid growth. Thus, in many cancer cells there is a frequent deregulation of Pol III in transformed cells. So, if the cancer cells have to divide rapidly, protein synthesis has to take place in a very high amount, and therefore more ribosomes are required, more transfer RNA is required, and therefore in many of these cancer cells the Pol III transcription is upregulated. So, Pol III transcription of transfer RNA in 5S ribosomal RNA genes as well as Pol I dependent synthesis of large ribosomal RNAs, this aspect we discussed in the last class, is very tightly linked to growth conditions. It decreases when cells are deprived of serum or nutrients, and it increases when cells are stimulated with mitogens. So, when there is a mitogenic stimulation, when cells have to divide, when cells are mitotically active, both Pol I and Pol III transcription go up, but when the cells have to differentiate or cells are going to quiescent state, the Pol III and Pol I transcription goes down. So, our Pol I and Pol III transcription is very beautifully regulated depending upon the need of the cells either to grow or divide or remain quiescent. So, the Pol III transcription of transfer RNA and 5S ribosomal RNA is known to be stimulated by oncoproteins such as c-MYC and repressed by tumor suppressor proteins such as p53 and Rb by directly targeting the essential Pol III transcription factor TF3B. So, because if the cancer cells have to, if the cell has to become tumorogenic, the cancer cells have to keep on dividing, they want more and more transfer RNA to be made, they want more and more ribosomal proteins and ribosomal RNAs to be made, and therefore, the first thing that happens in cancer cells is to upregulate the transfer RNA and ribosomal RNA synthesis, and therefore, the oncoproteins such as c-MYC as well as tumor suppressor proteins p53 and Rb play a very important role in interacting with transcription factors which are involved in Pol III transcription, especially the TF3B. Let us spend some time to understand how exactly this TF3B function is regulated by oncoproteins and tumor suppressor proteins involved in cancer. For example, as we all know, there are tRNA genes are organized in tandem units. There are several copies of tRNA genes arranged in a head to tail fashion in the genome, and normally in normal cells the amount of transacting factors such as polymerase 3 or TF3B or TF3C is limiting. Therefore, not all tRNA gene promoters are occupied by these transacting factors, and therefore, the level of transcription of the level of synthesis of tRNA is not maximal, because all the transacting factors do not occupy all the tRNA promoters. Only a certain level of tRNA transcription takes place, and this signifies the normal transcription, normal cell situation. So, pre-initiation complexes are actually assembled depending upon the availability of all these transcription factors, and it turns out proteins like P53, which actually are very important molecules regulating cell cycle, and very important mutations in P53 can lead to cancer. Turns out P53 goes and interacts with TF3B and makes it unavailable for formation of pre-initiation complex. So, in normal cells tRNA synthesis is regulated because TF3B molecules interact with P53, and as a result TF3B is not available for the formation of the pre-initiation complex, and TF3C transcription itself is rather regulated, and therefore TF3 is also not present in very high amounts, and therefore you only get a few molecules of transfer RNA made, just sufficient for the normal protein synthesis to take place. Now let us see what happens in cancer cells. In addition to P53, the other tumor suppressor protein RB is also known to be interact with TF3B. So, remember, in normal cells the tRNA synthesis is regulated primarily by making TF3B unavailable for the formation of pre-initiation complex in the case of Pol III promoters, because RB and P53 sequester the TF3B protein, and depending upon since these proteins are present in levels are very finely regulated, depending upon the level of P53 and RB, the tRNA level gene transcription is modulated. Now let us see what happens in the case of cancer cells. So in the cancer cells, the first thing that happens is the TF3C2, one of the components of TF3C is called TF3C2. As I said, I have not discussed in detail about the subunit structures of each one of these factors involved in Pol III transcription. TF3C is a very complicated transcription factor, consists of number of subunits, and one of the TF3C subunits is called TF3C2, and it turns out the TF3C2 subunit itself, which is actually the DNA binding component itself, the levels itself is upregulated in many of the cancer cells. So compared to normal cells, in cancer cells there are very high levels of TF3C, and therefore the number of tRNA gene promoters which are occupied by TF3C is much higher compared to normal cells. Finally, and also in many of these cancer cells, the cancer is because of mutations in tumor suppressor genes like P53 or RB, and when you have mutations in P53 or RB, they cannot interact with TF3B, and therefore they are not able to sequester TF3B and make them unavailable. Therefore, now TF3B is freely available for go and binding to the promoter regions of tRNA genes and initiate transcription, and in turn the TF3B now associates with oncoproteins such as ERK, CMYC and so on and so forth, which are growth promoters. Okay. So you can see in the normal cells, when the growth has to be regulated, tumor suppressor proteins like P53RB interact with TF3B and make them unavailable for tRNA gene transcription, and therefore tRNA gene transcription proceeds at a very slow pace. Whereas when cells become cancerous, because of mutations P53 or RB, these mutant proteins cannot interact with TF3B. On the contrary, the TF3B comes and interacts with oncoproteins which are growth promoting agents like ERK and P53. As a result of interaction, the tRNA gene transcription proceeds much faster, and therefore very high levels of tRNAs are synthesized. So you can see how cells have devised strategies for increasing the rate of tRNA gene synthesis by using either oncoproteins or tumor suppressor proteins, and this is how a normal cell will become cancer cell. It also turns out there is a very distinct compartment called perinuclear compartment, which is just on the periphery of the nucleolus. This is the nucleolus, and the dark body is the nucleolus, and the white body shown by red arrow is actually what is called as a perinuclear compartment. Now this component called as this subnuclear structure called perinuclear compartment was discovered in the 1992 and is present on the periphery of the nucleus, and this PNC is predominantly found in transformed cells of the cancer cells and is rarely observed in the normal primary cells. And in fact, the presence of PMC is often considered as a marker for distinguishing a cancer cell from a normal cell. So, in normal cells you rarely see this perinuclear compartment, whereas in the case of cancer cells this is very prominent, and it turns out this PNC is highly enriched in several Pol III RNAs as well as heterogeneous nuclear RNA protein 1 polypyrimidine track binding protein involved in hnRNA synthesis. So, you can see when a normal cell becomes cancer cells, in addition to the regulation of TF3B and increase in tRNA synthesis, there is also accumulation of Pol III transcripts and certain hnRNP particles in a perinuclear compartment, and therefore this becomes highly visible, and this PNC compartment is actually one of the markers to distinguish a normal cell from a cancer cell. A very interesting review article on the perinuclear compartment is available in this review article in Journal of Cellular Biochemistry just last year, and those who are interested in understanding more about this perinuclear compartment can read this article. So, in addition to this regulation of Pol III transcription in cancer, the Pol III transcription is also implicated in a number of diseases. For example, in the case of, as I mentioned in that long list of tables about the various RNAs which are transcribed by Pol III, in the case of neuronal cells there is what is called BC1 BC200 RNAs which are involved in the regulation of protein translation, and both BC1 and BC200 are transcribed by RNA polymerase III, and these are present only in the neural cells. Okay. And when you have problems in this BC1 and BC200 RNA synthesis, it leads to diseases like Alzheimer's disease or Fragile X syndrome. So, Pol III transcription is involved in neuronal disorders such as Alzheimer's disease and Fragile X syndrome. Similarly, the ribonuclease MRP, which stands for the mitochondrial RNA processing RNA, which is an RNA is involved in mitochondrial RNA processing, which is implicated in a genetic disorder called cartilage hair hypoplasia. So, you can see if there are problems in Pol III transcription or factors involved in Pol III transcription, it can lead to cancer, it can lead to a number of disorders, especially neural disorders such as Alzheimer's and Fragile X syndrome and so on and so forth. So, I think what I have described to you so far is about the general mechanisms or the major cis-acting elements involved in Pol III transcription, various transacting factors involved in Pol III transcription, what are the various types of Pol III promoters, type 1, type 2 and type 3, and what is the organization of these promoters, what kind of transacting factors go and bind, and how these factors in association with RNA polymerase III regulate Pol III transcription. Then we discussed very briefly how Pol III transcription plays a very important role in the normal growth as well as in transformation, and the synthesis of transfer RNA is very finely regulated. A number of oncoproteins and tumor suppressor proteins interact with the transacting factors involved in Pol III transcription, and as a result of these interactions either the tRNA gene transcription is increased or decreased. And when the tRNA gene transcription is increased, it can lead to cancer, whereas in normal cells the tRNA transcription takes place at a very suboptimal level, and not all the tRNA gene promoters are actually occupied by these transacting factors because they are sequestered by tumor suppressor proteins like p53 and RB. Now, we will spend maybe a few minutes to understand once the tRNA is synthesized what kind of processing takes place. Just like in the case of Pol II transcripts, we have seen the pre-messenger RNA undergoes a number of RNA processing steps like capping, splicing and polymorylation. Similarly, in the case of ribosomal RNA, we have shown it is synthesized as a precursor ribosomal RNA which then gets cleaved to 28S, 18S and 5.8S rRNA, whereas in the case of tRNA, let us see what kind of processing takes place. One of the first, there are about 4 major steps in the case of tRNA processing. One of the first steps is to removal by specific ribonucleases the 5' and 3' regions of the tRNA. You all know that the tRNA forms a typical structure called the clover leaf structure, primarily because of a secondary structure. The RNA folds itself immediately after transcription and forms a typical clover leaf structure, and a number of processing mechanisms takes place, and one of the first steps after the synthesis of tRNA, precursor tRNA, is the removal of the 5' and 3' overhangs by 2 ribonucleases called RNase-P and RNase-2. So, this is the first step in RNA processing, and once these 5' and 3' overhangs are removed, there is also a very short intron in the pre-tRNA molecules in the anticodon loop of the transfer RNA, and this intron is also gets spliced. So, removal of the intron from the anticodon loop is another important step in the tRNA processing. So, the 5' and 3' overhangs are removed, a short intron which is from the anticodon is spliced out, and then the the amino acid acceptor region called the CCA, which is actually added to the 3' end of the eukaryotic messenger RNAs, whereas in the case of prokaryotic tRNAs, it is part of the transcription. The CCA sequence is part of the transcribed RNA, whereas in the case of eukaryotes, the CCA sequence is added post transcriptionally. Remember, the CCA, which is very, very important for the tRNA function, is added post transcriptionally in the case of eukaryotes. So, is another important modification that takes place and known as the tRNA processing. And of course, we all know that just like in the case of ribosomal RNA, in the case of tRNA also, a number of unusual bases are present in the tRNA. So, this base modification also is a part of pre-RNA processing mechanism. For example, the dihydrouridine is present in the D arm, and the pseudouridine is present in the T arm, and a number of bases like cytosine and guanine and adenine, they all get methylated, and the first anticodon base sometimes is modified to inosine. So, all these base modifications also take place. So, the cleavage of 5' and 3' overhangs, the removal of an intron in the anticodon group, the addition of CCA in the 3' end of tRNA, as well as base modification, all these things happen once the tRNA is synthesized. Ultimately, you get the mature transfer RNA, which is then used for protein synthesis and translation. So, I think I will stop here. What I have listed here is, in addition to the articles which I have listed in between during the course of this lecture, I have also listed some very important papers which are very important key contributions for the RNA polymerase III transcription studies, especially from Bob Roeder's group, who made a very pioneering contribution, and I listed some of these articles, especially from Bob Roeder's lab. There are also many other important articles, but these are some of the key papers that one should read if you have to understand a little bit more greater detail the various aspects of Pol III transcription. I have also listed a couple of review articles which very nicely discuss about the various aspects of RNA polymerase III transcription research.